I'm Smriti. I'm part of the Combio department and I'm working with Sandeep. So my work involves finding out how moving out of equilibrium helps cells, especially in terms of transcription. So cells are the basic units of life and they perform a variety of functions, and they do this using specialized molecules called proteins. So cells make proteins through two processes, transcription and translation. So what we have is a stretch of DNA which acts as a template. Then the process of transcription creates an mRNA out of that template, and the process of translation converts that mRNA into protein. So there are two main processes out here, but one catch out here is that all the cells in our body have the same DNA, but they clearly have very different functions and they create different proteins. So the question is, how do cells know when they need to make a protein and when they should not? So this kind of regulation can happen at many levels, but the most prominent one is at the level of transcription. So transcription, this is a brief idea of how transcription works. So you have this strand, which is the DNA. You have the green part out there, which is the gene that turns into mRNA and creates the protein. And near the gene, you have this blue region called the promoter. So this promoter is a stretch of DNA that can bind molecules called transcription factors. So there would be an empty gene, a few transcription factors would come and sit on the promoter, and then the cell would decide whether or not to express that gene. So these transcription factors are of two broad types. They are either activating or repressing in nature. So for an activator, as you increase the concentration of an activator, transcription goes up, you get more mRNA. Meanwhile, if you have a repressor and if you increase its concentration, transcription goes down and you have less mRNA. So this is kind of like a molecular computer where this promoter region senses its surroundings, senses what type of transcription factors are present and how much concentration they are present in, and then decides whether or not to activate the gene. So and to begin studying this, we can look at a gene that has two transcription factor binding sites. So this is a simple diagram of a promoter, and it has two binding sites, the red and the brown one, and they can bind transcription factor A and B respectively. So this promoter can exist in 4 states. So 1 where there is nothing bound, 2 where only A is bound, 4 where only B is bound, and 3 when you have both bound. And you can calculate the probability of finding the promoter in any one of these states using StatMech. And to finally connect it with the transcriptional output or the mRNA, each of these states has a certain rate at which mRNA is produced, and there's a rate at which mRNA is degraded. And you have this equation out here that tells you how much mRNA you can expect, where these PIs are the probability of finding the promoter in any one of these states. So these kind of thermodynamic models have been widely used to gain a lot of insight into how bacterial transcription works, and they've been very successful in helping us create logic gates, engineer synthetic ones, and they were extremely helpful. But one thing that's important is that life requires energy. And recently there were a lot of papers that came out, and turns out that the equilibrium framework doesn't work so well in higher organisms. So there is a need to kind of understand how cells are using this energy and how moving out of equilibrium may or may not help them in certain ways. So the focus of my work is how would cells make decisions out of equilibrium. And in particular, I'm interested in decisions in the form of logical gates or logical operations. So we can extend the same model that I showed you earlier into a more general form, where you have the same 4 states, and these states can now transition between each other. So you have these clockwise transitions denoted by the blue arrows and the anti-clockwise transitions denoted by the red arrows. And when this relation between the rates holds, you satisfy something called the Kolmogorov criterion. And what this basically says is that if the rates are related in such a way, then this system is operating at equilibrium. And if this condition is not satisfied, that means the system is operating out of equilibrium. And since it's out of equilibrium, you need to put in some energy to sustain that kind of steady state it has. So that energy is given by that formula for delta mu, the chemical potential. So as before, the mRNA can be connected to these PIs, the probability of finding the promoter in each of these states. And since Ri and gamma are not dependent on these rates or the concentrations, we can only look at PI and get a good idea of how the mRNA production look like. So this is an example of occupancy profile and equilibrium. So out here you have these promoter states, the empty state, only A bound, both bound, and so on. X-axis you have the concentration of A, y-axis you have the concentration of B, and the intensity tells you the probability of the promoter being in that state. So for example, when both A and B are very low in concentration, you have a very high probability of being in the empty state. These profiles for equilibrium are always monotonic with respect to A as well as B. But when you move on and kind of move out of equilibrium, what you'll notice is that these occupancy profiles can look non-monotonic. And this has interesting implications. So for example, we'll take this profile and now we'll look at the mRNA production. So if we assume that A and B are activators and that this state is not active because there's nothing bound to it, and these three states are active, then we get an mRNA profile that looks like this when you have concentration of B and concentration of A. This is also non-monotonic. And if you if you look at the mRNA production along this blue line, you see this curve. So basically, this protein A, which is supposed to be an activator, is acting as a repressor within this interval. So it's changing its regulatory role. So this was interesting, but there were constraints to this. For A to alter its regulatory role, you need this cycle to be biased anti-clockwise, whereas for B, you need this cycle to be biased clockwise. So these conditions are contradictory, so you can't have alteration of regulatory roles for both proteins. And this has some consequences in the kind of gates you can and cannot make. So moving on to the logic gates, now that we have studied the occupancies and understood how the mRNA profile can be deduced using them, we found out that we were able to recreate NAND, NOR, or and AND gates out of equilibrium as well. But one interesting consequence of this non-monotonic behavior is that we get the emergence of two different logic gates. So this is the implication logic, this is A implies B, and this is the negation, which we've called the lack logic. So these rely on the non-monotonicity of the mRNA level with one of the transcription factors. Because if you wanted to create this gate at equilibrium, you would have to have an activator and a repressor. But out of equilibrium, the nature of the protein itself doesn't matter. So a consequence of the constraints about the non-monotonicity, since this alteration of regulatory role is mutually exclusive, we can't get gates such as XOR and equivalent. So this is the truth table for XOR. It is high when the inputs are mismatched. And you can imagine that to create a gate like this, we would need an mRNA profile to look like this. But this wouldn't be possible, right? Because because you have alteration of A as well as B. So yeah, so that brings me to the end of my talk. So to conclude, out of equilibrium, the transcription factors can alter their regulatory roles in a mutually exclusive manner. And that once, as a consequence of this alteration of regulatory roles, we are able to obtain two logic gates that can't be obtained very easily in equilibrium. And basically, you can use energy and create different kinds of logic operations, which would help the cell make different kinds of decisions. So I'd like to thank Dr. Sandeep Chaubey and my collaborator, Dr. Taya Middlecoop. He's based in the Czech Republic. And yeah,
Okay, thank you very much. Yeah. Any questions?
Hi, nice talk. So you said this XOR gate can't be, I mean, you can't reproduce the XOR logic with this construct or with this motive. So is it like you have seen other kind of motifs where, try to see whether other kind of motifs can give you this kind of XOR logic? Or is it something to relate, like the constraint is on the motif like that you are choosing?
So this is a very common motif that is used, but if you try to make it a little more complicated, then you could see XOR, like if you connect it to other cycles. But XOR is, they can be a little tough to make, and with this motif it can't be made. I don't know about the others, maybe.
Hi, nice talk. So I wanted to ask, is there a possibility that you have a state with incorrect binding? Like, or is it not allowed at all?
In this case, we haven't accounted for state with incorrect binding. It's a very minimal model, but yeah, in this case, it's not.
So I wanted to also, like, if you have an state with an incorrect binding, so the question is, even within that equilibrium picture, then would you see non-monotonicity?
No, I doubt it, because if we can somehow write the partition function for this, and if we won't get any kind of concentration square dependence anywhere, so it'll all be linear upon linear.
Okay.
So I doubt it.